Immunocontraceptives are an appropriate means of population control, particularly in urban and/or protected areas where culling is not feasible, desirable or legal. For contraceptives to be suitable in the field for wildlife management, it should render a high proportion of animals infertile for several years after a single dose and have nil or minimum negative side effects. The paper Long-Term Effects on Immunocontraception on Wild Boar Fertility, Physiology and Behaviour by Massei et al. Aimed to simultaneously test the injectable vaccine Gonacon for these two properties over a 4 to 6 year time period. The study was conducted in two trials between 2004 and 2010. In each of the trials there was 12 female wild boar of no fertility and each was randomly assigned to a treatment group. Trial 2 was designed to replicate Trial 1 and took place with 12 different female wild boar in 2006. Males were housed with both trials' control females for a time period of 3 months. Treated females in Trial 1 were housed continuously with a male for 4 years and for 2 3-month periods with treated females in Trial 2. The effectiveness of Gonacon to introduce long-term infertility was measured by collecting the immune response to vaccine, assessed by measuring serum antibodies to the GnRH, concentration of fecal progesterone, and reproductive output. Body weight was recorded every 6 to 9 months, parallel with blood collection, which took place at vaccination, 6 and 12 weeks after vaccination, and every 6 to 9 months thereafter. To determine whether the vaccine affected the social rank of wild boar, behavioural data was collected on interactions with trial mates. The data was compared before and after vaccination and between the two trials. All the results showed long-term effectiveness in infertility in most of the treated females. As shown, GnRH antibodies had an initial drop in the 12 to 24 months after vaccination, then stayed relatively but consistently low in the following years and was associated with infertility in 11 out of 12 treated animals. In Trial 1, none of the treated females gave birth in the 6 years after vaccination, and in Trial 2, high progesterone levels, as highlighted, indicated that 2 out of 6 treated females were cycling. One of these cycling females gave birth, while the other 5, including the second cycling female, did not reproduce in the 4 years following vaccination. The vaccine had no effect on aggressiveness, as both groups maintained their social ranks throughout the trials. Body weight increased with time in all groups. There was no signs of lameness in treated animals, and no obvious granitomia or abscesses were recorded at the injection site. In conclusion, the study showed that single-dose Gonacon introduced infertility for at least 3 to 6 years. It prevented animals from cycling, but did not affect their behaviour or physiology. The study also confirmed that injectable Gonacon met the safety and effectiveness criteria required for an ideal fertility control agent in wild boar.